Myself, Arshita. I'm going to explain about denaturation and renaturation of DNA. Denaturation of DNA means it is a loss of biological activity and is accompanied by cleavage of hydrogen bonds holding the complementary sequences of nucleotides together. This results in separation of double helix into the two constituent polynucleotide chains. That means the two helical standard native structure of DNA is converted to a flexible single standard denature state. The splitting of DNA molecules into its two strands during denaturation is obvious because the fact that the hydrogen bonds holding the bases are weaker than the bonds holding these bases to the sugar phosphate groups. The denaturation involves following changes: increase in absorption of ultraviolet light, decrease in specific optical rotation, decrease in viscosity. In increase in absorption of ultraviolet light, as a result of resonance, all of the bases in nucleic acids absorbs ultraviolet, and all nucleic acids are characterized by a maximum absorption of UV light at wavelengths near 260 nanometer. When the native DNA is denatured, there occurs a marked increase in optical absorbance of UV light by pyrimidine and purine bases, an effect called hyperchromicity. In decrease in specific optical rotation, native DNA exhibits a strong positive rotation which is highly decreased upon denaturation. In, de- in decrease in viscosity, the solution in native DNA possesses a high viscosity because of the relatively rigid double helical structure and long rod-like character of DNA. Disruption of hydrogen bond causes the decrease in viscosity. The effect of pH and denaturation. Denaturation of DNA helix also occurs at acidic and alkaline pH, at which the ionic changes of the substituents on the purine and pyrimidine bases can occur. In acidic solutions near the pH 2 to 3, at which the amino group bind protons, the DNA helix is disrupted. Similarly, in alkaline solutions near pH 12, the enolic hydroxyl groups ionizes, thus preventing the keto-amino group hydrogen bonding. The effect of temperature and denaturation takes place. The DNA double helix, although the stabilized by hydrogen bonding, can be denatured by heat by adding acid or alkali to ionize its bases. The unwinding of the double helix is called melting because it occurs abruptly at a certain characteristic temperature called denaturation temperature or melting temperature. The melting temperature is defined as the temperature at which of the helical structure is lost. The melting temperature is analogous to melting point of crystals since the guanine and cytosine base pairs has three hydrogen bonds as compared to two for adenine and thymine. It follows that DNAs with high concentration of guanine and cytosine might be more stable and have a higher TM than those with high concentration of adenine and thymine. In other words, the DNA molecules containing less guanine and cytosine bond denature first, as guanine cytosine bond has higher thermal stability. In fact, the melting temperature of DNA from many species varies linearly, with GC content rising from 77 to 100 degrees Celsius as the fraction of GC pairs increases from 20% to 78%. This measurement may force be used as an index of heterogeneity of nucleic acid molecules. Next, renaturation of DNA. Complete rupture of the two-stranded helix by heating is not a readily reversible process. If the solution is denatured, the prepared by heating is cooled slowly to room temperature. Some amount of DNA is renatured. Maximum reversibility 50 to 60% is usually attained by annealing. the denatured DNA. That is, the holding solutions at a temperature about 25 degrees Celsius below the melting temperature and above a concentration of zero point 0.4 molar sodium for several hours. Fast cooling will not reverse denaturation, but if the cooled solution is again heated and the cooled slowly, renaturation takes place. The restoration occurs because the complementary bases reunite by hydrogen bonds and the double helix again forms. The renaturation of complementary of single strands to produce fully double stranded molecule requires two separate reactions. First one nucleation reaction, second one zippering reaction. In nucleation reaction, the hydrogen bonds form between two complementary single strands, and this is called as a bimolecular, and also it is a second order reaction. In zippering reaction, the hydrogen bonds form between all the bases in the complementary strands, and it is also called as a unimolecular, and it is a first order reaction. This is the steps in the denaturation and renaturation of DNA. In to the native DNA, the heat is applied. It get denatured into two single-stranded DNAs. By cooling these strands, it forms renatured. That means it forms again double-stranded DNA. Thank you.